just about every scientific issue gets more complicated the more you look into it. And that, of course, is certainly the case with the next topic that we're going to approach, and that is genetic modification. And our speaker is Dr. Kevin Folta, who comes to us from the University of Florida, where he's a professor of horticulture. His main interest is actually studying the action of light on plants and how to use light to make fruits and vegetables more flavorful, how to give them a longer shelf life. That's his main area of interest. But he also is driven to educate the public on scientific issues. And one of the issues that he addresses routinely is genetic modification, which of course is controversial, and he has taken some heat for his talks on genetic modification, and he will explain to you why he has become a lightning rod for some of the anti-GMO activists. So Professor Kevin Folta is going to tell us some very, very interesting stories, not only about genetic modification, but about where it can lead when you talk about it.
Okay, thank you very much for having me in. You hear that okay? That sounds good. All right, good. I'm going to avoid the podium. I just would like to first say thank you very much for coming tonight. Thank you so much to Dr. Schwartz for the invitation to come to this symposium. Thank you very much to Emily Shore, wherever she is, for her outstanding organization of the symposium. And thank you so much to Dr. Lauren Trottier for allowing us this forum to be able to discuss science at the public interface. You're much cooler than that TED guy, whoever that is. So where we are is, let me go forward here, is I'd like to talk today about this issue of genetically modified food, or genetically engineered food, as I would prefer to talk about it. And I'll show you why in a little bit. I was going to just lead right into the talk, but there's been a lot of discussion lately about what's happening with me, some of the questions about my credibility as a speaker in this kind of forum. And so I was asked to give a very detailed disclosure about where funding comes from, where it doesn't come from, what do I do, and so we'll spend the first few minutes on that. My job is a professor, but also a chairman. I work as the chairman in the Department of Horticultural Sciences in the state of Florida for a land-grant institution, meaning that the faculty who work with me take care of the farmers in our state. We don't just take care of students, but farmers and also our citizens. And we have breeding programs that cover strawberries, tomatoes, blueberries, citrus. All of these are faculty in my department. We had the country's first organic and sustainable undergraduate program that teaches our students how to use these techniques and these production methods to raise foods with different environmental considerations and high market value for our farmers. We have plants on the space station. We have people like me who work in the area of genomics in order to use the most modern molecular tools to understand how to make things taste better and maybe how to use light to change the way plants taste. So we really very diverse faculty. My particular research program, I still have a laboratory and we still do a lot of cool work. Our main work, again, is in the light area. And currently the only funding my laboratory has is from a $500,000 USDA grant, which is to cover this particular line of work. Past funding in strawberry genomics has allowed us to not only sequence the strawberry genome in 2010, but also understand the genes that are associated with flavors, and that's the main driver. So there's a number of different national and state programs and some funding from strawberry-associated companies that have helped us to do this, and also many different countries where I've sponsored students to come and postdocs to work in my laboratory. I don't mention CONICET, which is another one that should have been on here, which is I've had a number of Argentinian fellows, and also internal grants and things like this. So funding from many diverse sources, zero research funding from Monsanto, never had, I gotta say it, never had research funding from them. I've had zero collaborations with them on research. They don't care about what I do. They're worried about corn and soybeans. I'm working with fruit. There's a cool picture of some of the research we do. So the other thing I do is outreach. And this is a big part of what I do. I love that interface with the public. This is something I do many times a week with physicians, with farmers, with scientists. And it's an important part of what I do because just keeping science in the laboratory is not enough, and we need more scientists to do this kind of outreach. And my program is called Talking Biotech, and the main thing I try to do is teach scientists to not be such scientists. I need them to start talking and being teachers. I need them to start talking to a concerned public that's worried about their food and worried about their health. And how do we connect with those people? And that's really been an important emphasis of my program for many years. And over the last 15 years, I've spent a lot of time talking to public audiences about the area of genetic engineering, talking about the good things, the things it doesn't do so well, maybe what's up in the future. All of that participation has brought me a lot of funding from different donors and different contributors. I had money from National Science Foundation to build lights that we used in classrooms to talk to kids about light and how light can influence plant growth. Last year, well, about 1 year ago, I met a employee of the Monsanto company at a conference who said, "Your students, everybody does so well. You guys do such great talks. What's the deal?" I said, "Well, we teach communication. We teach them how to talk to the public." And they said, "Well, this is great. Maybe we have some year-end money and we can sponsor your outreach program." And I said, "This would be fantastic because it would allow me," and none of this money goes to me, but it allows me to put out coffee and donuts at the workshop. It allows me to cover maybe an extra night in a hotel, things just so that I can do it. And so they gave us $25,000 to the University of Florida that I could use to write against for my costs associated with outreach. Now, when you're doing very compelling outreach for many years to talk to people about biotechnology and explaining it to them and make it not so scary, you get on the radar screen of many people who really don't want you to do that. And having a connection of dollars to your outreach program to Monsanto is exactly what they need to make your life quite miserable. And as part of this, and so here's some of the things that go on in social media. Monsanto pays for everything. He's just a propagandist. A tiny bit of money compared to what I actually bring into my research program. And Monsanto does not pay for everything I do. They gave a little bit of money to the university. We'll talk about that in a second. So what happened is that I was the first of 43 scientists to turn over 3 years of email to U.S. Right to Know, who's funded by the Organic Consumers Association, an organization that found it very disturbing that I was educating people about science. And so what they wanted to do was they said, "We want to find out how the industry is influencing your message. And so we need all your emails." And I turned over 4,600 pages of emails. I was the first to comply. I didn't even look at them myself. I really didn't care because I didn't think there's anything terribly weird in there. But however, when you give somebody who doesn't like you 4,600 pages of email, they can find a way to take a little bit here, a little bit here, a little bit here, and build the story they want to tell. Now, it's not the way we work in science, and so I was a little naive about turning them over, but I really did have to be a lot more careful about that. The story they told ended up in the September 5th New York Times, where if you read this, you find Eric Lipton told quite a story. He actually talked about how Monsanto funded all of my travel. And here's a graph that shows the travels funded by Monsanto during my career. Here's one. I had one travel funded by them last year when a group of farmers in Colorado wanted to learn something about this topic, and they didn't want to listen to the company. So the company called me and said, "Hey, would you mind doing this?" I said, "Sure, I'll do it, but I don't have any money." And they said, "Well, we'll cover the cost." So that's that. And so all of this really started into this entire this column and other fallout that really doesn't represent me accurately. And what's really disappointing about that is that I'm incredibly transparent, I'm proud of my funding record, and I'm excited to show you where the money comes from that allows me to do what I do. And now you can even go, I've spent about many hours to put together all of this on a page where you can go through the last 2 years of every seminar, every talk, every thing I've written, who paid, how much they paid, how much I was reimbursed. And what you find is that I lost a lot of money personally. I mean, it costs me money to do things because I don't always get reimbursed. Sometimes I lose receipts. And so I lose money by being in science communication, and maybe I get reimbursed if I'm lucky. Most things are done for free because that's my job as a land-grant scientist. I work for you, or at least those south of the border. I work for them. I work for you too, though. And so that's my real job. And I'm very transparent about this, and I hope you look here because I really want you to see that. I have nothing to hide. I'm the most transparent guy out there, and I want to set a standard so that the anti-GM people will also adopt that same level of transparency. Don't hold your breath. So here's what happens. Because I've participated in this discussion, I'm Monsanto's brain and Monsanto's shill and Monsanto. Here, the world's most discredited professor and getting this money, and he's a lying whore. Actually, here they tell you that I got the money to promote GMOs, which is nothing further from the truth. Then they said here, hilarious, they're putting it on Craigslist in his town. This is nuts, it's hilarious. Well, here they had my deceased mother, they put on there, on her birthday, so they knew when they were doing this. They put my address, they put the address of my laboratory. There were veiled death threats. There were quite obvious threats. My boss of my university, when I spoke at Cornell, had to have the FBI terrorist task force involved because of threats against me, because I teach science. And this is where this gets real serious. They took back that money. My university did. They moved that $25,000 away from science outreach, where we teach kids about citrus greening and teach kids about light, and move that money to a food pantry, which is great. But that's money out of what I used to use for outreach because of the fallout from this. And what's really sad is that not only have journalists defamed a teacher who just wanted to talk about science and share with the public, now other scientists won't do what I do. They won't go out and talk to the public. And worse, even today, I was supposed to talk to CBC, and they canceled because they said, we got a call that said that we shouldn't hear from you. So not only are scientists not participating in this discussion, and students are being scared away from it, the outlets where we would discuss it are not accepting a scientific message. This is a crisis situation, folks. And this is being brought about by fear and other issues which are being catalyzed by this debate around genetic engineering. So that long introduction now will lead to what I would like to present to you today. That's very nice. But wait, there's more. Never missing a pile to jump onto. The food babe Vani Hari on InfoWars, where you can buy oxygen in a bottle, and then she can sell you an antioxidant on her website. She also has requested all my emails, and so she will be receiving those as well. And I just wanted to mention that there's a book coming out called The Fear Babe, where I've written the foreword, and this will be coming out October 29th. So, and this is really the point, is that now we have people who are going after a teacher because I'm costing them money, and I'm just teaching the science. And now I want to walk you through that today, because no matter what you feel about transgenic crops or this idea of genetic engineering, whether you're for it, whether you're against it, whether you're on the fence, I really want you to challenge your thinking on this. And I want to take you to a different place in the way you think about it, because I think about this very differently than most people, because I've been studying it since I was a 10-year-old kid who took a book out of the Downers Grove Public Library on recombinant DNA technology. Nerd. And I've been studying this forever, and I get it, and I want to share this with you so you do too. And please, let this be our introduction. Don't hesitate to email me, contact me, phone call me. I know your emails will be part of a FOIable record. But the point is, I want this to be an introduction. I want to be available to you, and I want you to understand this, whatever it takes. Okay, so here we go. I guess the first question I'd like to ask is by applause. How many people think that genetic engineering as a technology has many more benefits than risks and is generally a good thing? Okay. And how many people think that this is, by applause, is generally more risky than beneficial and probably pretty negative? Come on, a few of you out there, right? Yeah, that's good. Don't be shy. It's kind of sad. I can go home now. How many people, and this is always the biggest group usually. This is a scientific audience maybe, so maybe a little different, but how many people are just not sure? Okay. And that's good because you're the folks I would like to reach the most, and you're the ones I'm shooting for here. The folks who really like this technology, I'd like you to think about ways that I communicate this and think about ways you could incorporate this into the way that you talk to people about it. So, I wanted to ask you this first. Just yell out for me real quick. When we talk about food and food sustainability, food and food systems, what are your concerns? Anybody? Antibiotics. What else? So herbicides. So food safety issues is kind of where we're going there. Food safety. What else? Price. So food safety and price. And what else? I'm sorry. I'm sorry? Nutrition. Okay, that's a good one. Sustainability. So we're talking about environment, we're talking about safety, we're talking about quality. And any audience that I ask in the developed world, in the industrialized world, tends to tell me this. I'm psychic too. So you guys really did fit exactly what this audience says. Now let me tell you what's important to me. Or better yet, let me tell you what's important to the Alliance for Science Fellows who come from all over the world from 10 different developing countries. When I ask them, they say, we're worried about the developing world. We're worried about people who are starving. We're worried about food safety and farmers. We're worried about the environment. And we're not so worried about the people on the wealthy side of the planet. When you ask me what's important and how I prioritize this, it's very similar. To me, I worry about those folks who have nothing. It's been a really emotional time lately, so excuse me. For me, why do I put my feet on the floor every morning and go do this? And why do I have, this stuff on the internet about who I am and what I'm about? This is where I come from. And this is why I want you to understand who I am and where I'm coming from, because why I want to remodel the way you think about this question. I'm thinking about serving others. I'm not so worried about taking care of myself, and I'm not so worried and I'm really worried about how we can have more sustainable farming here in the States, in our environment, in the United States, not here in the States. So we have a good situation. Our food supply is the most plentiful it's ever been. Fewer people have to be employed to make this food supply. The average age of the North American farmer is 58, and very few people coming up behind them. But it's the fact that a few people are willing to do this work that 99% of us don't have to do it. And that we can pursue things like leisure time, that we can study other things, we can have other jobs. And this is what's really set up the problem we have, is that we have this new distrust of food. Now let me hit you with a couple concepts and then we'll talk about them in detail. Right now we know that humans have always participated in plant and animal genetic changes. We know that this technology is a precise extension of breeding. We know, and this is a great quotation, that the techniques we use pose no more risk than traditional breeding. And that reminds us that even when we're rubbing plant parts together to create a new kind of plant, a new variety, there's some risk there. In 18 years since we've been doing this, there has not been one case of illness associated with a transgenic plant or a GM plant or plant product. And we don't make any transgenic animals for consumption yet, and there's really just 10 traits. 3 traits in 10 commercial crops in general. Now the problem is that when you have new technology and you have something like the internet, you now create a situation where people can do their own research, and really the only experts are the ones online, and experts like me don't really matter anymore. We've been marginalized by too much information that isn't always of good quality. And so how are we going to fix that? And what we have to do is we'll zip through this today in about the next half hour. Lots of material to cover. We'll talk about how we do this genetic improvement, about where our crops come from, how our current traits work, because if you understand the current technology, it's not so spooky, and what are the things we didn't do and how we go forward. And I really just want you to rethink this entire question. I think that you'll be surprised where we come from. So humans have been genetically improving crops for a long time, and what we get are better yields and maybe better products. We get improved nutrition, maybe grow in other places where they previously didn't grow. And so plant genetic improvement has been something that has gone with the human explosion of population and really allowed us to have civilization. But if you ask a general audience what plant genetic improvement is, they have a very different idea. It's something very sinister, something maybe we shouldn't be doing. But what exactly are we doing when we're genetically improving a plant? Well, let's talk about genetically improving animals. What's natural? And here's broilers from 1957, and this is the number of days after they've been grown. And you can see how breeding and human intervention in the genetic improvement of chickens, in this case, produced a much better product with much better commercial variety, much better commercial quality. And that's just in something like broilers. If we look at plant materials, or crops, we can look at this in a slightly different way. None of this, or very little of it, came from North America. And this map is a really interesting one because it shows the centers of origins of all of our different crops, or many of our different crops. And what you find is that citrus came from Southeast Asia, that onions are over here by Pakistan, alfalfa from the Middle East, almonds from the Middle East, apples from Kazakhstan, sugar beets, lettuce. Everything that we eat was from somewhere else, and now is a foreigner on our soil. And this is important to remember because this was human mediated. And what I'd like to get you comfortable with is the idea that genetic changes and changes in plants are sometimes mediated by humans, actually more often than not. Nature didn't give us anything very good. That what we have and the incredible bounty of the most healthy and wonderful food supply in human history is because humans intervened. Some evidence for this is in this slide, and I do a whole talk on crop domestication that maybe we'll talk about some other year, but the idea is that the ancestral forms of our crops, this is corn in its great-great-great-great-grandparent, and modern-day corn. And this took 16,000 years, and many generations of someone identifying the one plant that didn't die, and then keeping those seeds for the next year, planting those seeds, keeping the one that had a little higher yield. And through time, that created this much better version of this antecedent. But we see all the tremendous improvements of our fruits and vegetables happening in all of our different areas, whether it's seeds that have been removed from bananas by adding chromosomes, or tomatoes, and all the varieties of tomatoes that come from these primitive forms that are almost horticulturally useless. Humans have intervened in changing plants, and we've done it in some really wild ways. The next thing I'll show you is a table that is really complicated, but I'll explain to you what this is about. And if you Google Frankenfood Paradox, you'll find this. What it tells us is that we make plants in different ways. We do genetic improvement in different ways. We can make hybrids, where we're just crossing plants. We can make polyploids, where we artificially double or naturally double chromosome numbers. We can do what's called mutation breeding, where we add either chemicals to seeds or use radiation to damage DNA and create genetic variation. We can cross species barriers, crossing plants that would never cross in nature. It may be cases where we have to go out and pull out the embryo out of the seed and put it in culture in order to get it to grow. So weird, but we do that. All of that. We have no idea what genes were changing. We have no idea what genes were moving. We have no idea how many genes we're affecting. No clue. Nobody tests. Nobody cares. Nobody wants to label it. Certainly nobody's too worried about it. That's what we've done forever, moving tens of thousands of genes at a time. Now let's talk about the technology that we use in genetic engineering. Here, we're moving one gene, maybe a couple others. We know exactly what it does. We understand it. We can trace it. We understand exactly how it works. And yet, this is the technology that people are afraid of. The thing that we understand, the surgical version, is the thing that freaks people out. Whereas all of this wild west of plant genetic improvement, moving whole chromosomes and tons of genes at one time, nobody really cares. And to someone who's a kind of an aspiring plant breeder and someone who knows genomes, it just doesn't compute. Humans can have the opportunity now to induce these precise changes that we understand that have much more safety. And we can do that. This is what you see online when you're asked about how this process is done. And there's nothing further from the truth. For one, we don't use a syringe or anything like that. The whole process is based upon this bacterium called Agrobacterium tumefaciens, which is nature's genetic engineer. It's able to move a piece of its DNA into a host plant cell and kind of sets up shop inside that plant cell. It's a natural infection. is what this thing does. It moves a piece of DNA. And Agrobacterium does this, and the process is very complicated. But what the idea basically is that you can use the bacterium to move the gene of your interest into that plant cell. It's done by a little bacterium that does this process. And then you take that one cell and you can use culture to grow it into a whole plant. Now, I just told you in 2 minutes what would normally take an entire lecture. But the idea is that this is a very well-established process of using a bacterium to install the gene into a single cell that we can now grow into an entire plant. So these are the current crops that are available. And I think people are surprised to see things like wheat not on here. It was even on Joe's slide last night that there's wheat. So there's like corn, soy, alfalfa, cotton, potato. These are newcomers that aren't quite available yet, but all of these, at least these 4 are in the human food supply. These are limited acreage. So these are the crops that are available now. And the 3 traits that are really available we'll walk through very quickly: virus resistance, insect resistance, and herbicide resistance. Because I want you to understand how these work, because when you understand how they work, they're not nearly as scary. So first we'll talk about virus resistance. And virus resistance works because of a process called RNA interference. And RNA interference is a pretty cool process, and it's a natural process in a cell that when a cell gets a virus, certain kinds of virus, it'll turn on a process that will degrade the genetic material of that virus. So if you're able to put in a gene that you want to silence, like let's say a viral gene, if you put in some sequences, you can then target that gene to turn it off, or turn off that virus. And I know that wasn't very clear, but what the general idea is that you can introduce a construct you can introduce to the plant, engineer in, a copy of the virus in a way that it essentially cancels out the virus that's infecting the plant. And when you do that, it allows the plant to grow. The nice part about it is that these are sequences that are naturally there upon viral infection, and that it really cuts the needs for insecticides to control the insects that vector virus around the plants. The bad news is that there is a problem with pollen containment. If you go to Hawaii, there are growers who would like to grow non-GM papayas who sometimes have trouble doing that because of pollen drift. And that's a consideration we always need to keep in mind. Oops, I lost a few slides there, but we'll go ahead and talk about Roundup Ready. So Roundup Ready is the other area that people are very interested in. And Roundup Ready really is a way of a farmer to plant something like soybeans. And as the plants start to grow, you can use an herbicide over the top to kill all the weeds, but leave the soybeans still growing. So the herbicide kills everything except for the plants that have been engineered to resist it. And I'll tell you about how that works, because it sounds kind of scary, but it's actually pretty simple. Now, if we look at a biochemical pathway, and think about this as some product being converted to some product, being converted to some product that ultimately ends up as amino acids, which are the building blocks of proteins. There's an enzyme in plants called EPSPS that catalyzes that step. It takes B, turns it into C. But when you use glyphosate, this compound that's in Roundup, glyphosate binds to the EPSPS enzyme, and it blocks its function. And when you block that function, the plant can't make amino acids and it dies. Now, bacteria have a very similar biochemical pathway that uses a slightly different form of that enzyme. And when you take that enzyme and you put glyphosate on it, it doesn't care, doesn't bind it. So now you see how simple this could be. What if you could take this one and put it in the plant? Then that plant, in theory, could grow in the presence of the herbicide. And that's exactly how this process works. That the plant enzyme is essentially, well, we add the bacterial enzyme into that system, and now that plant lives just fine. It doesn't bind glyphosate, and the plant now can survive. Nothing terribly magical. It's almost like changing your light bulb, throwing it away and putting in one of those LED ones. It's a more efficient way of doing exactly the same thing. And now allows these plants, gives the farmers a very powerful tool to be able to use less time and less labor in being able to grow their crops. The advantage of this is that farmers can now apply a very low toxicity herbicide that is cheap and works well. The disadvantage is that you start to grow weeds that become resistant to this technology, and that's what's happened over the last 15 years. That when you're using such stringent selection, you're killing all the weeds, the ones that can survive now propagate. And things like palmer amaranth make 500,000 seeds and they propagate like wildfire. And one of the downsides of this technology that we're seeing right now is the presence of glyphosate-resistant weeds. And the heat map here shows that the hotter colors represent greater numbers of these weeds. And this is one of the big challenges and certainly a downside of this technology. So I would like to use the rest of the time today to talk about success stories we can't use, because this is really where I want to focus. This is what I want you to think about when we talked about the things that we share as our common values: to take care of people, to take care of the planet, to take care of the farmers, to have food that's safe. That's stuff that we all want. We can do that. Those technologies exist, but we can't use them because of a complex regulatory system and a public that is afraid of this technology. So I need you to dream with me and understand how we can use this to fix the things that we care about. Because when we can do that, we can move this technology forward. So let's talk about some of the things we can do. The papaya ring spot virus is already done. This kind of got a little bit out of place here. But this was where we used this RNA interference. These have the transgene. Oh no, these do. These don't. And you can see the difference in the survival of the plants. The next slide shows this really well. Here are plants that are genetically engineered, surrounded by those that aren't. And you can see how the virus just destroys the vigor of that crop. Now we can apply that same technology to some other plant species as well. The other place that we can do this is in biofortification, and golden rice is the first example. Golden rice is an example where you can use the genes from something like carrot to be able to make rice, which is white, produce beta-carotene, the golden component of carrots. And what this allows you to do is now be able to produce rice that could alleviate, at least in part, some of the vitamin A deficiency, which kills between 250 and 500,000 people a year. Something that definitely would be an interest in the developing world. And the same technology has been applied to cassava, which is a vigorous root vegetable that is a staple in Africa. A lot of this goes to waste because of cassava virus. And in this case, cassava brown streak virus decimates African farms. And we can fix this with the same technology used on that papaya. This is being opposed by Greenpeace. This can be done and should be deployed. Similarly, in Africa, the use of vitamin A-enriched bananas, beta-carotene-enriched bananas, that now people who have bananas as 70% of their calories can survive and have enhanced vision because of the presence of this trace nutrient in their food. They need to get less than a tiny fraction of a grain of sand a day of this stuff, and they don't get it, but here they can get it. And again, people standing in the way of deployment of this technology. And this isn't Monsanto, folks. This is the governments of Uganda, of Kenya, of Nigeria, the Gates Foundation. Folks here who are trying to do the right thing, yet people standing in the way. We need to de-Monsantoize this conversation and talk about the way we can solve the problems we want to see solved. And this is my podcast here, Talking Biotech. If you want to listen more of this, I do interviews with these folks who, very fascinating people. They also have a problem with bacterial wilt, that bananas die because of bacteria that decimate the banana crop. And part of the problem is that you don't have good antimicrobial controls in places like Africa where you can spray them and control the bacteria. Yet there are a number of genes that can be installed into this plant, in the banana, that make them resistant. And some nice examples here, that this is the genetically engineered ones. This is the one that's not genetically engineered. And you can see the difference. And if you're a farmer in Africa growing bananas for your family or other families, and you have to make this crop work, this would be extremely valuable. And it's not just a one-gene solution. There's different genes that do this very well. So we wouldn't have the same kind of threats of resistance like you would see in glyphosate and resistance to weeds. This could be a very durable tool, and they're pushing this as hard as they can towards deployment, yet people are standing in the way, saying that genetic engineering is dangerous and that this is going to harm people. In Africa, they tell the men it's going to make you impotent, tell the women it's going to make you infertile. They're scaring them away from this technology. In the industrialized world, children walk around in school with EpiPens because of allergies to peanuts. Here's an example where science has been able to suppress the major allergens in peanuts, turn them off. It's been done. Why don't we see it? Because it costs money to commercialize, and no industry is going to do it because of the high costs and because of no public acceptance. This is something that could save lives. It's not being used. We have chickens that have been invented back in 2011 that don't get bird flu. And this year in the American Midwest, they bulldozed 45 million birds in the landfills because of this virus. We can stop that virus just by having these birds that are genetically engineered with a cool trick that doesn't allow them to transmit it. They get sick and die, but it's not contagious. Super cool. We could do this, but we don't do it, again, because of the lack of acceptance. We have salmon that can grow to market weight in half the time. And if you think about the early effects of high protein diets and good protein diets for children, this could make fish proteins more accessible to the young people in our countries and worldwide. Again, none of these are the big ag companies. These are small startups. This fish was made in 1989 and still has not been released for human consumption, although all the tests have been done for years. It's a political decision. It's a political decision. It's not a decision made on science. We know pigs that can be engineered to have lower phosphorus output, which is tremendous for our environment. We have potatoes that will grow and produce less of this chemical called acrylamide, which is a carcinogen and a neurotoxin that is produced by potatoes when you fry them. These new potatoes that are now approved don't produce those levels of acrylamide. They also don't brown, they don't bruise, which means that a farmer has the same amount of land can get a much higher yield of marketable potatoes because they won't have to take out the bruised ones, which Americans won't buy. So again, it's something it's helpful for the environment and helpful for the consumer. Non-browning apples, again, another technology that makes it more likely that the food that we pick from that tree gets into a human because it's not going to turn brown when you cut it. It can be used in more applications and more recipes. In Florida, we have this thing called BS2 tomato, which is a pepper gene put into tomatoes. They're cousins on the evolutionary tree. And what it allows us to do is, you can see the difference between the genetically engineered ones and the non-engineered ones, which have a very bad bacterial disease. And you can see it better here. These are not genetically engineered, these are genetically engineered. And the control for this disease is currently copper. And so conventional and organic farmers use between 6 and 10 pounds of copper as a pesticide on these crops. And they have to do it usually weekly. So huge amounts of a heavy metal going into our environment. And packing houses for tomatoes have so much copper in them that it has to be treated as toxic waste. This gene from a neighbor, a pepper gene in tomato, would allow us to grow these plants more sustainably without those inputs. It saves money for farmers. It's easier on the environment. Why aren't we doing it? Because of fear. We have grapes that can grow in Florida. We have a BT brinjal, which I didn't talk about BT, but this is an anti-insect protein which allows these to be grown in Bangladesh, and they're doing very well there at this point. Normally it's children who are spraying pesticides with no protective equipment in Bangladesh, and this allows them to not have to have that exposure. Yet there are people who are standing in the way of this as well. We can engineer healthier oils into things like canola or cotton that allow us to not have to depend on fish for high nutritional value oils. In my state, all of our trees citrus trees are dying. Most of you may not know about this, but in Florida, we have something like 90 to 100% of our groves are infected with a disease called Huang Bong Ling, or HLB, or citrus greening disease. This is what it looks like. The trees are just dying, and they're dropping all their fruit. We have genetic engineering solutions. But how do you go from a genetic engineering solution that takes 10, 13 years to approve, and then make 60 million trees? Trees, not little plants. And then get them up to production. Our industry is in a state of peril, and we needed this back in 2005. Yet we're waiting for the solution still, because the industry is afraid of a backlash from the consumers. In my lab, we made strawberries that don't require fungicide. And so fungicides are very prevalent on strawberries. Here, these are treated with fungus. These are treated with fungus. These have a gene turned on all the time that helps them resist. It's a plant gene that helps them resist the fungal infection. And so you don't have to use fungicides. This seems brilliant, but our industry doesn't want it because they're afraid of boycotts. So who's blocking the solutions? Where does the fear come from? And what it comes from is a very highly paid level of people who are making a lot of money by getting in the way of science. who are requesting my emails because I come here to tell you about tomatoes and oranges that can help benefit from this technology. The big problem, and a lot of this is anti-corporate, and one great quotation by Hank Campbell is, these people hate corporations more than they love people, and I think there's a lot of truth to that. So we'll talk a little bit about myths, and then we got to wrap it up. Maybe I even have to skip the myths, but we'll talk briefly. This is one of the most famous images, are 3 rats that they say link genetically modified food to tumors. This paper originally came out in 2012. There's 3 rats, one that gets GMO, which no scientist would ever put the word GMO, it's not a scientific term, so I'm curious there. GMO plus Roundup and Roundup alone. And then these rats are miserably tortured animals that are exceeding any level of animal laboratory ethics. If this was in my lab, I'd be shut down. You don't let this happen to animals. What's missing? Control. What about the rat that didn't get GMOs or Roundup? Well, if you look on table 2, he got tumors too. So this is a very compelling figure that has been used in the anti-GMO movement. And it's been used to scare people into resisting the cassava for the people in Africa, the oranges for Florida, to ask for my emails because of this. And you see the problem we have here, folks, is that we're getting bad information, and it's stoking fear and making a very compelling narrative. I had some other things I wanted to zip through here, but I think I'm going to skip that so I can get to the end. We know that right now we have the safest food supply in human history, yet we're being misled into being suspicious of it. And it's being fomented even more for a profit, and this bothers me a lot. Companies like Chipotle say, a farewell to GMOs, just because if you can guarantee that a huge part of the population is afraid of this, whether it has any merit or not, but then say, hey, guess what? We don't have that over here. Now you're making a brilliant business decision on horrible science. And the funny part is, their soda still has high fructose corn syrup from GE crops, and all of their cheese is made with microbial rennets, which are generated in genetically modified, probably fungus with a camel gene. So they still are using GMOs, they just don't tell you that. So I'm going to wrap it up here. We got a couple minutes. The last thing I'll talk to you about, how do you talk to people about this? And when I talk to different audiences, I like to give them a take-home assignment. The mistake we've made as scientists, and I did it for 12 years, was talk to a public audience and do this wrong. And it's because activists key off of emotions. You have people who aren't experts who are using the messages of fear to influence the majority of people on the center of that curve. Scientists are coming at this not by saying, let me tell you how this is right or wrong or why it relates to what you care about. We're coming at it and saying, guess what? Well, this paper, and this paper, the statistics show this with 95% consummate, and people turn off and they don't care. Scientists have been screwing this up. The companies have screwed this up. How do we fix it? We have to be, as scientists, speaking to the folks in the center of the curve, not wasting our time down here. And the other things we have to do are avoid some simple mistakes. Get rid of this feed the world rhetoric. Our farmers feel very passionate about feeding the world. They say they feed the world, but it's a throwaway concept. It's a throwaway line. It's cliche. We don't feed a world. We're trying to feed his world. We're trying to feed that family's world. We're trying to feed that town in Africa. These are specific examples that I can point to ad nauseum, and I can introduce you to people in those places who can tell you how this technology could change their lives. We need to be specifically speaking to these specific instances. Always discuss the strengths and limitations. If you're going to talk about Roundup resistance saving money for farmers, you have to talk about how the weed resistance is coming. It's how you have credibility as a speaker. We never claim it as a single solution. Scientists don't say biotechnology is going to solve everything. What we say is that it's a part of a plan, that we will use genetic engineering in oranges along with better nutrition, insect control, improved genetics, other tricks to solve that problem of citrus greening. And the other problem is that it's very attractive for people who are in the area of biotechnology to bash ideas like organic or sustainable, organic production, where I don't go there. There are two different ways of solving a problem, and actually they work really well together. We're all looking for sustainable farming here, and that we have to keep that in mind. This is just a tool that we can use to fix the things we care about. It's really important that we remember that. So farmers are taking action. They're getting very involved in social media. And if you care about this topic, I hope you do too. Right now, it's dominated with bad information, and we need more credible voices in that space. Excellent opportunities online for you to integrate. And I left conclusions open because it depended on time, and so I got maybe a minute, so I'll conclude with a couple things. What I really hope to impress upon you today is that this is not Monsanto's technology. This is your technology, it's my technology. Don't let them have it. Why don't we think about the things that we can do with a very safe way of moving a gene from one place to another in a very predictable and very precise way? We need to lower regulations. We need to empower our academic institutions. We need to have more funding for research in the public sector. Let me play in this space. I would love to compete with Monsanto. I'd probably win. The trick is to give us more options. Right now, it's only a place that they can play because they're the only ones with the money and the lawyers and the know-how to get through it. We need to have a wider dialogue and a better dialogue, especially with those that oppose the technology, to make sure that we're implementing it safely and correctly. And that's best done by those of us in the public sector scientists that aren't really Monsanto prostitutes. You know, I want to do this, and we all need to be on the same page for that to happen. So if you listen to the podcast, I think it's really good. It's driven by excellent guests. I just ask a question now and then, and I hope you listen to it. It's really good. And the last thing I'll conclude with is if you don't know the name Norman Borlaug, I want you to go home and Google him tonight. And sorry. It's been a really bad month. You know, I put my feet on the floor in the morning for the same reason he did. How do we use technology to solve problems we care about? How do we solve problems for people? How do we fix this? And this is why we need to have a better conversation around this topic. I want to wrap up with one quick thing. How many people who were on the fence before we came in feel a little bit better about this technology? Okay, well then my job is done then, and I will look forward to your questions. Thank you very much.